Hello friends, good evening. I hope you are doing well. And today the topic which we are going to discuss is Human Genome Project. And the tagline is that it is completed. And this topic is very important and very knowledgeable for UPSC Aspirants, UPSC, any state PCS examination, NEET examination, CSIR NET examination for the life sciences and current affair purposes. So let's see that what are the updates in Human Genome Project. So introduction, basically the global collaboration called telomere to telomere project. So let's we take the laser pointer here so it can have a better understanding. Means what was left in that previous Human Genome Project which was announced in 2003. So basically you know that around 2 decades ago it was incomplete and about 8% of the human DNA was left unsequenced, because that 8% genome was termed as junk DNA. That 8% genome was termed as junk DNA or repetitive sequences, or which is now known as heterochromatin. Scientific paper published recently, a large team has completed the remaining 8%. Means this was remaining at that time. Now the scientist has completed this remaining 8% of this genome sequencing, which is a new addition. And the pictures of the human genome for the first time has been released. Genome sequencing, we will take a brief account. Laboratory method that is used to determine the entire genetic makeup of a specific organism or cell type. Then why the scientists are behind and they are after to sequence the human genome? Because this can help to understand how specific disease such as cancer arises and genetic changes during the course of cancer and the precancerous conditions, diagnosis and the disease. Now we will see that what is the procedure for genome sequencing. What is exactly genome and genome sequencing? Genome is the entire set of a genetic material. Means is suppose in a human being the entire 23 pairs of the chromosomes, you know that is known as that genome. Okay, so in the first step, that DNA shearing means, as the name itself indicates, that the excision of the DNA cannot be done by the simply seizures. The restriction enzymes required for this, which is composed of millions of the bases, you know, and the adenosines, cytosines, thymonosines and guanosines, they cut into the pieces that are small enough for sequencing machine to read. Means they are cut into the small pieces. Then the second term comes that individual fragment has to be tagged, or that is known as the DNA barcoding. Addition of DNA tags or barcodes to identify which piece of seared DNA belongs to the which bacteria or organism, especially in the case of human genome. This is similar to how a barcode identifies a product at a grocery store. Okay, now genome sequencing. The machine, the barcoded DNA which is prepared now from multiple bacteria or multiple organisms or a single human are combined and put in a whole genome sequencer. There are many whole genome sequencer. Now advanced versions are also coming into the market, and the genetic sequencing advancing very fast. The sequencer identifies the adenosines, cytosines, thymogens and guanosines or bases that make up each bacterial sequences. Now the entire sequence has been read by that particular sequencer and it has been sequenced, we can say. Now the main thing in the next generation sequencing or genome sequencing is the data analysis. Okay, the scientists use computer analysis tools. They use the tools and they require very high sophisticated instruments or computers, very high memory they are required. Then the bacterial sequence and identify differences. Means they analyze that what differences and what they will analyze into a meaningful way that this can be a probable genetic map. The number of the differences can tell the scientist how closely related the bacteria are, and how likely it is that they are part of the same outbreak. Means suppose you are going to analysis of this human genome, then the entire sequence will be put into the computer, that will be multiply aligned, and then it can be adjudged that this particular sequence belongs to bacteria, humans, or something else. So on the basis of the data analysis in next generation sequencing, the data analysis is the main part. Then we will take a brief look at the timeline of Human Genome Project, that what is the Human Genome Project, how it was started. So in 1996, the commencement of the Bermuda Principles, which is related to the gene sequencing or genetic material. Then in 2000, the draft Human Genome Project was released. In 2000 also, the genome browser were also released to analyze the bioinformatics analysis. Then in 2022, the genome-wide association G was studied, because now the human genome was with the scientists, so they can play with the genetic sequences, and multiple tools and techniques came into the market. So, and multiple projects also, that project is that they are known as the genome-wide association, means how genetic tool is different, they can study. So in 2003, the first human genome was released. Then 2005, first commercial next generation sequencing services launched. You know that in 2005, the next generation sequencing was very costly, and it was not a easy cup of tea. The scientist has to work hard for that next generation sequencing, to do the next generation sequencing. The sequencing was probably easy, you might go, but the chemicals were very costly, and there was a lack of softwares and the tools. In 2013, the Phenome-wide association, that was known as the PHWAS, was launched. Then after 2016, the launch of Human Atlas was also done. But now finally in 2021, including that 8% of junk DNA or heterochromatin, then now 100% the scientists are claiming that in 2021 the final human genome map is released. So what was not sequenced? What was not sequenced? So you will be curious to know that what was not sequenced. First sequence, you know that in 2003 from the Human Genome Project it was released. Then the Human Genome Project was an international project which started in the 1990 and completed in 2003. Only euchromatin part was sequenced, which consists of 92% of the entire genome, and the heterochromatin was not sequenced. You know that what is euchromatin and what is heterochromatin. Euchromatin is the part of that particular chromosome which is continuously involved in the proteins production and sequence, and the heterochromatin is generally silenced, and that is assumed that they are not actively participated into the protein formation and transcription and the translation. Heterochromatin is a small portion of the genome and does not produce code or proteins. There were at least two key reasons why heterochromatin was given lower priority. So anyone can ask that why it was not sequencing earlier. So the scientists completed nearly 92% of the sequence. What was that? They can also sequence in that 8% also at that time. But there are the probable reasons. Reasons for not sequencing heterochromatin is that the first is that this part of the genome was thought to be junk. Junk DNA means it was reported that it is the junk DNA. Junk means it is useless and it is non-functional. But now it has the importance of this junk DNA has been realized, and the scientists have proposed the 8% of that genome sequencing, and the entire sequence has been the human genome has been sequenced. Now the second, the euchromatin contained more genes that were simpler to sequence, and that was a limitation at that time. You know that there were no sophisticated instruments at that time. So simply the euchromatin was the main target of the scientist at that time. The fully sequenced genome is the result of efforts of a global collaboration called the telomere to telomere project. Means now that has been that project which has completed this 8% of the genome sequence. The project is known as the telomere to telomere project. You know that in chromosomes telomeres are the lateral part or very distant part, so they are not easy to sequence. So what is unveiled? You will ask that, sir, what is unveiled? T to T CHM13, highly repetitive DNA sequence found in and around the telomeres. Means when that this 8% genome is sequenced, then highly repetitive DNA sequences and around the telomere that are found. Structures at the end of the chromosomes. You know that I told that telomeres are the end structure of the chromosomes, and the centromeres. Centromeres is also at the middle section of the each chromosome. You know that the centromere is at middle and it plays a vital role in the cell division. Reveals long stretches of the DNA that are duplicated. Means it was initially also believed that it is very duplicated sequences and repeated sequences are there in the genome and known to play an important role in the evolution of the disease. Means this particular 8% of the genome plays a vital role in the evolution and the disease, and it revealed a large number of genetic variations, and these variations appear in a large part within these repeated sequences. Many of the newly revealed regions have important function in the genome, even if they do not include active genes. We know that, and the scientists also know that that this particular region is not involved in the active functioning, but still the genes which are found and which has been analyzed in this particular project are known to play active role. Then recently, in 2 and 3 years, past 2 and 3 years, the governments and across the globe, not only for India, across the globe have realized the power of that life sciences or NGs. In 2020, Ministry of Science and Technology had announced an ambitious gene mapping project, which is known as the Genome India project. Inspired from that human genome project, the central government have also launched Genome India project that indicate that to promote a Indian genome that can be easily accessible, and this genome can be used for that treatment of the specific disease, genetic disease. The mega project aimed to form a bank or gene bank, we can say, after collecting 1,000 samples in the first phase. Means initially the government is planning to collect the blood and to collect the DNA of only 10,000 people in first phase, and that it will execute at large scale if it will be very useful. Then the addition to the available information, why government is spending so much of resources on that? Because they want to add the available information on human species and advance the cause, both because of the scale of the Indian population and the diversity. They want to study that which diseases are prevailing, which genetic diseases are prevailing, which genetic structures are prevailing. So to know about the genetic imprint, the government is planning to Genome India project. In recent 2021, India formed a Indian SARS-CoV-2 Consortium on Genomics, which is known as a INSACO. Means a consortium of 10 genome sequencing labs across the India in December 2022 to pump up efforts for genome sequencing. You can see that the government is also doing efforts at the war scales to promote the genome sequencing. So you can understand the importance of genome sequencing in modern era. So with that, I will conclude my today's topic. We will meet on some day of the week, and if you are new to my channel, then please subscribe my channel, like my video, and share if you like it. And if you have any suggestion, then you can also give it into the comment box. I will certainly try to implement on my next presentations. Thank you very much.